Hi, and welcome to this overview of Cytobank. My name is Jeff Cracker, and I'm an application scientist at Cytobank. Cytobank provides the leading end-to-end, cloud-based, compliant, and secure single-cell knowledge management solution. There are several parts of Cytobank. First is the high-dimensional analysis and structured content management pieces of our software in the cloud. We have tools like Spade and VisMe, and I'll cover more later, that are in that component. We also have this structured content management, which is comprised of things like inbox searching and collaboration tools. These are part of our software offering. However, we also have an informatics consulting service. So if you have questions about analysis, if you have a need to develop some SOPs, or to analyze a large cohort of data that you've already collected, we'd love to help you out with that. So here's what we're covering today. We'll talk about a summary of Cytobank security and tools, our high-dimensional analysis workflow with VisMe and Spade, looking at automated discovery with Citrus. I'll take a look at the Cytobank API, and also how Cytobank handles data management. Cytobank is designed around security. Our servers are hosted on Amazon's cloud, AWS, and you can see several other details here about the security practices we entail. If you'd like more details, they're available here at this URL. Cytobank is a fully featured cytometry analysis program, from traditional analysis techniques like histograms, dot plots, compensation for fluorescent data, things like statistics export and heat maps. All those are standard in Cytobank, but we also offer these advanced tools like Spade, VisMe, Citrus, and Sunburst, in addition to advanced ways to interact with this data, like the Cytobank API, which I'll be covering later. So let's take a second to look at what a complex phenotyping workflow might look like. First, we'll briefly identify some populations and create some sunburst plots. We'll annotate some figure dimensions and describe how those affect the figures, create the heat maps using those figure dimensions. We'll look at some VisMe results, and then we'll look at results from VisMe data clustered with Spade. Here you can see our traditional gating interface, where you can draw gates, either histograms or two-parameter dot plots in this plot. We've already gone through and gated some of these standard populations, and you can see down here a population sunburst generated from these traditionally gated populations. And this is a great way to get a really quick look at what changes between populations and how your hierarchy is laid out. So it's based around circumference of a circle. So ungated is all of your events, so it's 100% of their circumference. Singlets is, in this case, just a little over half. So it's only half of the circle's circumference in an arc. And the further away from the middle you get, the deeper into the hierarchy you're looking. And so say you want to focus only on CD3 positive cells. You can click on CD3, make that 100% of the circle, and examine where all these subpopulations fall. And then you can select whatever file in your experiment you'd like and see how things change. From here, what we can do is hop into the working illustration. This is kind of central to how Cytobank works. We take the populations that we've generated using our gating interface. We've annotated each of our files with how they were treated using conditions. In this case, it's a pretty simple experiment, but if you have more complex experiments, you turn on other experimental dimensions and annotate those. And again, we use these so you don't have to work with complex or unclear FCS file names. This tagging system is in place to give scientific context that's great for collaboration, exploring the data efficiently, and searching. And then using those figure dimensions and rearranging them in their order up here, again, this is central to how Cytobank builds figures, you can generate any kind of plot type you'd like. In this case, we're looking at heat maps, and you can see very briefly that the IL-7 stimulated samples, and this CD4 T cells, react by increasing the STAT5 or phospho-STAT5 expression. And if you want to see what that looks like instead of just yellow, you can mouse over this cell and see what the stimulated instead of the unstimulated data looks like. So now it's a great summary tool, but up until now it's been traditional analysis methods. From here we can jump into VisMe results. VisMe allows you to group similar events into two dimensions using information from many channels in an automated manner. This allows you to gate on the results like you would on normal cytometry data. Let's hop into the software to see what this looks like. In case you're unfamiliar, each of these dots on this VisMe plot represents an event from the original data files. The closer two dots are in these two parameters, tsne1 and tsne2, the more similar they are across the n dimensions that you use to set up the analysis. So from here we can see by coloring each of these dots by their expression levels of a third parameter, in this case CD4, we can see what the phenotype is of each of these islands in this VisMe plot. So in this case, this is the CD4 positive island. This is the CD8 positive island. And as we scroll down, we can get a better idea of kind of what's going on inside each of these. Here we're looking at CD45 array, and you can see the positive to negative spectrum across both the CD4s and the CD8s. And it's then possible to draw a gate, through the middle of one of these islands if you prefer. It also works for rare populations. You can see things like CD34 positive cells. This is a bone marrow. You could draw a gate around this little peninsula up here to isolate those. And go through and just make sure you have a good grasp on where all of these populations are. So VisMe is a fantastic tool for finding rare populations or for showing relationships between subpopulations. From here, we can hop into SPADE on VisMe. We've got this data so far, but when you run SPADE on these parameters, it essentially sets SPADE up for success. And you can run SPADE traditionally in Cytobank too. You don't have to run VisMe first. But in this case, this is what it looks like. Here you can see we have a traditional SPADE tree, but again, on this VisMe data. So we also have our tsne1 and tsne2 parameters. And now when you color the SPADE tree by things like, right now, CD34, if you click on these CD34 positive nodes, you can see that it mirrors where our population sat in our original VisMe plots. And the same holds true. So if you click on CD8 positive, it lights up the CD8 positive islands and CD4 as well. And we can color those by CD8 and CD4, so it's easy for you to see. So this is a very powerful tool to look at and chain together the best of both of these algorithms, the rare population identification of VisMe, and you can look at the fold change comparison in SPADE then on that VisMe data. We also have Citrus, and I've mentioned this a few times now. Citrus is another kind of dimensionality reduction algorithm, except it's geared towards a little bit different kind of data. Citrus allows you to get statistically significant comparisons of large groups of samples instead of just a few samples at a time. So the way it works is it essentially clusters your data automatically to identify rare populations, similar to how SPADE goes about clustering. But then it uses learning algorithms to identify the clusters and cell response features are the best predictors of a known experimental endpoint. This essentially tells you, based on 2 experimental groups or more, which features are different between those 2 groups. You can either choose the strongest features or the most predictive features, or you can choose all the features below a specific significance set point. So it's a very powerful tool for looking in an objective way at large groups of data. So in this case, this is an example of Citrus data. You can see there's several kinds of results it generates. One is a tree that is similar to a SPADE tree. And you can see here it's identified this node as having the CD20 positive expression. So these are our B cells in this node here. And then after doing its comparison step, it's also identified that same node as being different between the 2 groups we had in this experiment in phospho-S6 expression. We can then take that node number and look for its full phenotype. So you can see CD20 positive, 3 negative, it's CD7 negative, IgM positive, and HLA-DR positive. So it's pretty obviously some B cells here. So it's a great way to say, all right, given this data, what changes between my groups? In addition to our advanced analysis algorithms, we also have an API which lets you access functionality from the command line. So this is geared more towards computational biologists or bioinformaticians, However, we don't want to scare anyone away. It's actually a very powerful tool for using Cytobank without accessing Cytobank through the browser like a normal user would. So instead of going through Chrome or Firefox or Safari, you can essentially remote into Cytobank and either send batched instructions to Cytobank to spin up a SPADE or a VisMe run, to upload data or download data. Or instead of using Cytobank's tools automatically online, you can download data, run your own custom code on your local machine, re-upload the results from that code, or attach them to an existing experiment. And then Cytobank will also talk to existing LIMS systems or central repositories using this same technique. So we have a way to interact with your system if you have an institutional-wide backup or data storage system as well. And lastly, Cytobank provides robust data management tools. So hosting all of this data is great, but it doesn't do much for you if you can't find it. So the inbox in Cytobank provides great tools to organize, search, and share your data. So we have what are called projects for group-level sharing if you'd like to give a selection of people access to a selection of experiments. You can search in your inbox on names of experiments or names of people. You can search for reagents used in each experiment. So if you know you used an anti-FOXP3 or phospho-STAT5 antibody, you can search for only experiments that have that antibody in them. Or you can search based on channels. You said, you know what, I know I had a PE size 7 CD38, and I'd like to see only my experiments with that channel in it. You can do that as well. And lastly, you can share experiments just one-on-one by giving somebody full access. You can see here a project that I've created. We have some leaders, some members, and they all have these levels of access to these experiments. So again, it's a great way to share with groups of people at your institution or with reviewers who'd like to see the raw data, but you don't want them to change anything. Projects are perfect for that. We also have, again, in our inbox, ways to search for experiments based on name or top sample tags by pulling out, only experiments with IL-7 or unstimulated, or channels, only things with phospho-STAT5 or Ki67, things of that nature. So as a whole, Cytobank allows you to go from raw single-cell data, do all the annotation steps, the scaling, the gating you need to, as a scientist, make that available to reviewers and collaborators as you get more advanced analyses and summary figures completed, and then automatically share that with any directors or managers you have. And then anybody who you share these experiments with can then go back and double check on the original compensations or scales or gates if they want to. So it's a very powerful tool enabling collaboration across the institution. So how do we stack up to the competition? Cytobank allows you to access your data from wherever you are, as long as you have access to a browser and an internet connection. Cytobank is cross-compatible across many different platforms, both PC and Mac. The updates are applied automatically. Both desktop software and Cytobank are secure. Cytobank has increased tools for collaboration between users because the data is hosted on our servers. You don't have to worry about moving the files around once you upload them. Cytobank has a structured search component, which allows you to search by figure dimensions or by channel names, in addition to author's name or experiment name. Our compute is scalable, so as you start more analyses and need more compute power, we automatically add that. We also have traditional analysis tools, and we're always expanding our advanced analysis algorithms. In conclusion, Cytobank provides exceptional analysis, collaboration, and archiving tools on a secure and flexible cloud platform. So if you have any questions about Cytobank, please visit our information portal at support.cytobank.org. There you can write us an email or look up our knowledge base, or you can email sales@cytobank.org and ask us. Thanks so much, and have a good day.